Alright, hello everybody. This video is going to be about your macromolecules of life, and we are actually going to split it into two parts just so it's not too long of a video. So the way this next few days are going to go is we're going to cover the first two macromolecules, and then we're going to start some practice, and I would encourage you to focus on the practice that we covered today. And then tomorrow we will wrap up the macromolecules and cover the last two. You're going to do some more practice, and then you're going to have a whole day to work on the macromolecules assignment and to continue practicing. Before we actually get started with the notes, I just want to go over a couple of things that are going to be important for you. Number one is this lesson here is what your essay is over. And so in your study guide you have a chart, and that is the same chart that you're going to see the day that you take the test on biochemistry. The chart is not going to be on the quiz. The chart is going to be on the test. And so what you'll do is you'll just fill in the chart, and as long as you have prepared, you should be good to go on that. It's going to be worth like 16 points, so it's definitely something you want to prepare for. And as we go through the lesson, I'm going to point out the things that you need to be putting in your chart. So I would definitely pay attention to that as well. Now, the second thing is the quiz over this unit. You're going to have a quiz over two sets of notes. It's going to be over chemistry and water. And so on that set of notes, it's going to be probably more vocabulary-like. You're going to have, you need to know the parts of the atom. You need to know definitions like what a compound is, what a molecule is. You need to know the different types of bonds, the properties of water. And so when you're studying that set of notes, you just need to be sure that you understand the material. Some will be definitions, some could be true, false. Just make sure you're familiar. Now for this particular set of notes, there's only going to be one type of question on the quiz. And every single one is going to have the same four answer choices. And that's going to be carbs, lipids, proteins, or nucleic acids. And so hopefully you did a little review activity after you finished the water notes the other day, just to kind of test yourself. And on each one you had a card, and you were supposed to identify whether it fit in carbs, lipids, proteins, or nucleic acids. Your quiz is going to be the same way. I'm going to give you a statement, and you're going to answer which group it goes in. And so just kind of as an example, it could say DNA and RNA. And when you see that, you should know that that fits under nucleic acids. Or it could say made of amino acids, and that would be proteins. So you're not going to have to give me specific information about the macromolecules. You just have to recognize which statements go under each of the macromolecule groups. Okay, so maybe that'll help you in your studying, and I would encourage you to make sure that you feel really comfortable with the material. And so really, try hard on your practice and ask questions if you have them, but that's how you need to prepare for the quiz. All right, so let's go ahead and get into the note material. And so we're just going to start off with just a quick review. So first off, we have organic compounds. And so back in your chemistry notes, we know, hopefully, that organic compounds are made up of carbon. It's going to be carbons attached to hydrogens. And normally these organic compounds are going to be relatively large. And that's what we're talking about when we say macromolecules. So that prefix macro means big or giant. And so in terms of molecular size, these things can be huge. You could have a molecule of water that has a whopping 3 atoms. In the case of DNA or RNA, you've got, millions of atoms that are bonded together. And so it's just important that you understand that in terms of molecular size, these things are really big. But all of them are going to be carbon compounds. They're all organic compounds that make up living things. And so 3 of the 4 are what we refer to as polymers. In fact, on your essay, one of the columns is asking for the monomer that makes up the polymer. And so 3 of the 4 are polymers. And so that's going to be your carbs, your proteins, and your nucleic acids, okay? Now, what it means when we say polymer, remember that poly means many, it's made up of many repeating subunits. So it's kind of like links in a chain. You have a repeat of a link attached to a link, attached to a link, attached to a link, and that's going to give rise to the chain. So each individual link would be the monomer, and the whole chain would be the polymer. And so we've got all different polymers. Plastics are a polymer, a spider web is a polymer. And the key is you have smaller repeating units. Now, the one that is not a polymer are lipids. It's not that they're not big, they're still a huge molecule, but they don't have a repeat unit like the other three do. And so when you get to lipids on your essay, you're just going to name the parts that make it up rather than having a monomer that repeats. Now, these polymers are going to be built by a type of reaction called dehydration synthesis. And I always like to break words down. And so if we take this term here, let's first look at this word, synthesis. Okay, the word synthesis means to make or to build or to put together. And so anytime you synthesize something, you're making something. And then the first word, dehydration, if you're dehydrated, you're losing water. And so essentially what's happening in a dehydration synthesis is a bond is formed. Okay, we're building something, so we're making bonds. A bond is formed, and when that happens, a water molecule comes out. And so this literally means lose water to make. Okay, and that's how I would remember it. In a dehydration synthesis reaction, we're losing water to make. And in the case of these big polymers, we're making covalent bonds where those electrons are shared. So I just kind of want to show you where that water is coming from. So here's an example here. These are monosaccharides. They're just little sugars. And when they come together, notice you have an OH and an HO there. When they're joined up, there's just an O left. And that's because those two H's and one of those oxygens came out in the form of water. And so down here, every time that bond is made, a water molecule is lost. If we kept adding these little monosaccharides, we'd lose another water molecule, and so on. And so in order to build the polysaccharide, or in order to build the polymer, we're going to lose water as those covalent bonds are formed. Now, hydrolysis is going to be the opposite. Okay, literally, hydro means water, lysis means to break or burst. And so in this case, we're going to add water to break. So dehydration synthesis, we lose water to make. Hydrolysis, we add water to break. And so if we wanted to break down like a protein or break down a polysaccharide in digestion, water molecules are going to be added to break those covalent bonds. And so these are just the two main types of reactions that are going to occur as we're building polymers and breaking polymers. All right, so now we're going to get into our actual macromolecules. So today we're going to focus on our first two groups, which is going to be carbs and lipids. And so our carbs, a lot of times people know that word because we hear about carbohydrates in our diet. I think carbs are delicious. And the reason why is because carbs are sugars that give us energy. A lot of times I repeat myself over and over. If I were teaching in front of you, I would probably say that about 5 more times. So I'll just go for a couple. Carbs equals sugars equals energy. Okay, carbs equals sugars equals energy. That's what you need to know. And so the reason carbohydrates are so important, and the reason you do need to eat carbohydrates, there are good and bad ones, but you do need to eat them, is because they are our main energy source. Now, just to kind of tie this into some future lessons, we perform a process called cellular respiration. And in that reaction, we take carbohydrates, especially glucose, and we break it down in the presence of oxygen, and we use that to make ATP. So that's why we say that carbs are our main energy source. Now, there's a couple other functions, and we're going to look at these more as we get into our different types, but there's energy storage. Okay, I do want you to add something here. Please always add to your notes anytime something is said that might be helpful. Normally I would draw on the board, but I can't really do that. And so it's going to be kind of hard to write. I've tried it, but I'm not very good at writing. But what I want you guys to do here under energy storage is I want you to write short term. I'm just going to abbreviate it. Okay. So the energy storage that carbs are able to do is a short-term energy storage, okay? We're going to see another macromolecule that stores energy, and it's not going to be short-term. And then another function here is structure, and we'll take a look at that in a moment. All right, so there's 3 main groups. Prefixes are pretty important here. We have monosaccharides, disaccharides, and polysaccharides. And saccharide is just a fancy name for sugar. Okay, so anytime you see that SACCH, that's going to be a sugar-related molecule. You may have heard of saccharin. It's one of those other forms of sweeteners. So this is a single sugar or one sugar. This is a double sugar or two sugars. And this is a polysugar or many sugars, okay? And so we're going to take a look at each one of these. Now, the monosaccharides, if we're talking about our essay, the monosaccharide is going to be the monomer for your carbohydrates, because these are just one little sugar. Some examples are glucose, which is probably one of the most important sugars that we're going to talk about in biology. And the basic structure of a monosaccharide is they have this 1 to 2 to 1 ratio. That's because you have a carbon, there's your organic compound, bonded to a hydrogen and a hydroxyl group. And so this is one of those functional groups that we looked at. This hydroxyl group will make it polar. It's one of the reasons that your sugars will dissolve in water. And so what we're talking about here is just the general formula. This N could be any number, okay? Now, for example, glucose is a 6-carbon sugar. So the formula for glucose would be C6, and then you double the H's, H12O6. All right, so all of your basic monosaccharides, for the most part, have this formula. Ribose over here is a 5-carbon sugar, so C5H10O5. And then their structure is typically this ring type structure. So you can see this is a 6-carbon, there's a hexagon there. This would be a 5-carbon, so you see a pentagon. I do want to remind you that you could have isomers. So some of your sugars have the same formula. They're just arranged differently. And so just know that that could give rise to different shapes and slightly different functions. Okay, even though they may have the same formula, like glucose and fructose both have C6H12O6, they're not exactly the same thing. That's what we call isomers. And so it's your monosaccharides that are the main energy source. They're the major nutrients for our cells. That's what we use to make ATP. All right, our next group are our disaccharides. Once again, di means double or two. And so in this case, we have two monos that are going to be joined together. Now this is going to be a dehydration synthesis reaction that happens, which means that covalent bond is formed and a water molecule comes out. And so the covalent bond that we're dealing with in carbohydrates is called a glycosidic linkage, okay? A glycosidic linkage. This is one of the things you need to know for your essay, because one of the columns are the specific types of bonds that you will find in those macromolecules. And so it is a covalent bond, but we need to be more specific in that in carbohydrates, in your disaccharides and your polysaccharides, it's a glycosidic linkage. It's just a fancy type of covalent bond. And so that's what you're looking for in that bond column. All right, so you can see here that you put two monosaccharides together. Some examples are sucrose, that's just your basic table sugar, the white sugar that you might use in baking. You've got lactose, which is a sugar in milk. And you've got maltose, which is in like malt beverages. And so these guys are held together by that glycosidic linkage. And these are also going to be just energy for our cells. We can break them up pretty easily. And then the last group are your polysaccharides, and poly means many. So in this case, we could be talking hundreds or thousands of monosaccharides joined together. Once again, we have that bond called a glycosidic linkage. And so depending on the monomers, which sugars are making up the polysaccharide, and depending on the positions of the bonds, we get different structures, which means different functions. And so here's 4 examples of polysaccharides that you need to be familiar with. We have starch and glycogen. Notice both of these store energy. So earlier when we were talking about short-term energy storage, these are the two that we're talking about here. So starch is how plants store energy, and you've probably eaten plants that are starchy, things like potatoes or rice or grains. And then glycogen is how animals store it. And so like we have big chains of monosaccharides. This normally happens in our liver. When we need energy, we can break them down, but they do store them for us short term. And then the other two, cellulose and chitin, were that third function. It talked about structural components. And so these are the primary molecules in cell walls. So for cellulose, you'll find those in plant cell walls. For chitin, you'll find it in fungal cell walls. And so once again, they're just big, long chains of sugar, but they make up cell walls. So when it comes to your essay, you're going to have to name the macromolecule. That would be carbohydrates. You have to give the monomer, okay, which we talked about what that was. In carbohydrates, that would be a monosaccharide. You need to mention the bonds, and then you're going to have to give an example and what they do. And so you've got several of those that we've already talked about. All right, so here's some examples of what those polysaccharides might look like. Okay, cellulose is what we actually call fiber. We have trouble breaking it down. And then you've got glycogen and starch, those energy storage molecules, and the energy stored in all those chemical bonds. All right, the next group we're going to cover are lipids. And our lipids, remember, were the non-polymers. They're still huge, they just don't have a repeat unit. And so one of the things that groups them all together is that lipids do not like water. And to tie this back into our chemistry lesson, the reason they don't like water is because lipids are nonpolar, or they typically have nonpolar regions. And nonpolar and water do not mix, okay? So lipids are a group of nonpolymers that are hydrophobic. They do not like water. And the reason is because they have a lot of these big hydrocarbons, which are nonpolar compounds. They have several functions as well. We've got storing energy, okay, and this is where I want you to add long-term. So the carbs were short-term energy storage, the lipids are long-term energy storage. And these guys can store tremendous amounts of energy. There's lots and lots and lots of covalent bonds. And when we need them, we can break down those fat reserves to get energy. They are going to be fats and oils, and so fats are going to help insulate us. And then we're also going to look at a special lipid that makes up our cell membranes. So the ones that are going to store long-term energy are going to be your fats and your oils. And this is where on your essay, it's talking about the parts. So remember, we don't have a monomer, but we do have parts. And so your typical fats and oils are going to consist of a glycerol. Okay, that's this portion here. All right, that's a glycerol backbone. And then in the case of fats and oils, they're going to have 3 fatty acids. You can just put fatty acids as the parts. You don't have to designate a number. But this is where we get the term triglyceride. We have 3 fatty acids attached to a glycerol backbone. And so these guys here are your long fatty acid tails. You can see all those carbons, and you can see all those hydrogens that are around them. These are the portions here that do not like water, okay? And that's what makes these fats hydrophobic, are those fatty acid tails. Now these guys can store tremendous amounts of energy because you see all these lines. Those lines are chemical bonds, and bonds store energy. And so when we break those, that's how we get the energy from them, okay? Now, to tie into your essay, we have a bond. Now, once again, it's a type of covalent bond, but in lipids, the bonds that are going to be holding these fatty acids to that glycerol are known as ester linkages, okay, ester linkages. When it comes to your triglycerides, we have 2 different types of fatty acids. We have saturated, which means they're saturated with hydrogens, okay, and the reason why is they only have single bonds. So to kind of go back, you see how there's hydrogens all the way around. Every single one of these is a single line, single line, single line. And so literally these fatty acids are saturated, or they're completely full of hydrogens. So carbon can form 4 bonds. And in this case, because they're all single bonds, we get a saturation of hydrogens. And so what happens is it kind of creates that very rigid structure. And so these are the ones that are solid at room temp. These are going to be your animal fats. Now, the other type are unsaturated. And the reason they're unsaturated is because they have 1 or more double bonds. And so when that happens, it causes the fatty acids to kink, okay? And when they're kinked, they can't join up so uniformly, and that's what causes them to be a liquid. So here's an example of saturated. Notice there's all single lines there. You got hydrogens going all the way around. You have that more rigid, solid type structure. Whereas over here, when you throw a double bond in the mix, notice there's no hydrogens down here, okay? So there's still hydrogens, but it's not saturated all the way around, and that's what causes that kinking to happen. And these are going to be your liquid, your plant oils, the ones that are healthier for you. All right, another very important lipid is a phospholipid. And so we're going to have a similar structure to the triglycerides. We're going to have 2 fatty acid tails, okay, instead of 3. We're still going to have that glycerol backbone. So once again, in terms of your parts that make up lipids, you can talk about a glycerol and fatty acids. But we have, in addition here, we have a phosphate group, okay, which is where we get phospholipid from. And there's going to be a polar region. And so what's unique about the phospholipids is that they're what we call amphipathic. They actually have 2 different regions. They have a hydrophobic region. That's going to be those tails that we've already looked at. But they also have a hydrophilic region, and that's what we call the head of the phospholipid. And so this area here is actually water-loving, okay? And so this gives rise to the basic structure of our cell membrane. Phospholipids are incredibly important because they're the main structure in our cell membranes. And so what happens is cells are aqueous. There's going to be water inside the cell and outside the cell. And so what happens is the basic structure forms because those tails that do not like water, remember these fatty acid tails are hydrophobic, they turn away from the water. And so the heads that like the water go on the outside, and the tails that don't go on the inside. And that's what gives us this bilayer. So phospholipids are pretty important because they're the basis for our cell membranes. And the last lipid that we're going to look at are steroids. Okay, now they obviously don't have the same structure. We don't really see fatty acids here. They have 4 fused rings. But what puts them in the group of lipids is they still are hydrophobic. Okay, they don't have a monomer and they're hydrophobic. So that's why they're in this group. A couple of important steroids, you've got cholesterol, that's going to be in our cell membranes. It actually helps kind of stabilize cell membranes. And then some of our hormones, especially our reproductive hormones like testosterone and estrogen, they're going to be a type of steroid. And so once again, they're lipids, so they're hydrophobic. And that is going to finish up our first lesson on macromolecules. So I would use this time that you have left to start working on the practice, maybe focus on the carbs and the lipid section. You could also maybe take a peek at that essay and start thinking about what you're going to put in those blanks. And I hope you guys have an awesome day.